All right, class. So this week we're talking about our last category of macronutrients, which are proteins. There'll be two videos this week, with this first one talking about the building blocks of proteins, the monomers, and the various types of proteins that we get in our diet. So proteins are large polymers of amino acids. Many amino acids come together in particular sequence and order to create a wide variety of proteins. Proteins are arguably the most important, or at least the most impactful macronutrient in a living thing, because these are the tools of the cell that essentially make everything else happen. Okay, so in order for our cells to be able to get energy from carbs or lipids, a protein is needed in order to transfer energy from that molecule. In order for us to digest our food, a protein is needed. In order to break those molecules apart, in order to build more molecules, a protein is needed in order to do that. So proteins essentially take care of everything that is happening in the cell, or at least control everything that is happening in the cell, because they are carrying out the instructions from our genes. And they do this based on which amino acids are used to build them. And so amino acids have nitrogen in them. So whereas carbohydrates and lipids were just composed of carbon, hydrogen, and oxygen, amino acids do also have nitrogen in their amine group. And this is the primary source of nitrogen that we get in our diets. So these large polymers of amino acids have 20 different types to choose from. Right? So you can think of these as 20 different tools to use, or 20 different material to use to build some structure. We have 20 different amino acids that all living organisms use to build proteins. And so the general structure of an amino acid has a central carbon. And from that central carbon, we have 4 other groups branching off of it. So every amino acid will have an acid group, which is this carbon with oxygen and a hydroxyl group off of it. This is our acid group. Another bond will be given to an amine group, which is our nitrogen. A third bond will be given to a single hydrogen. And then the fourth bond is what dictates what type of amino acid that is. So this is our R group, or our side chain. And again, this last bond is what will change between amino acids. So a glycine, for instance, just has another single hydrogen attached to it. A leucine, excuse me, very sorry. A leucine has this chemical structure with lots of carbons and hydrogens. And aspartic acid has a couple of carbons and some oxygens and a hydroxyl group, right? So whatever is taking up this fourth bond, making this side chain, is what determines which amino acid this is. And each amino acid has slightly different chemical properties to it. So glycine is neutral. It does not carry a charge. Leucine is hydrophobic. It does not dissolve in water. Aspartic acid is electronegative, so it is carrying a negative charge. And thus, an amino acid like aspartic acid could make some magnetic attractions with positively charged amino acids or positively charged molecules and structures in the cell. So that chemical structure, that chemical property, is what ultimately leads to the role and usage of the protein that it helps build. Right. So we have 2 different types of amino acids to consider. We have our essential amino acids. And so just like what I introduced with essential fatty acids in the last chapter, the term essential does not necessarily mean it is more important than any of the other amino acids, but it is essential because we cannot synthesize it, and so we must get it from our diets. So really this term is referring to the fact that it is essential to consume. We must eat it. We must get it from some other organism because our bodies cannot synthesize it, our cells cannot synthesize it. And then we have non-essential amino acids. So these are the amino acids that our cells can synthesize. And we would do this by transferring an amine group from one amino acid to a different one, or to another acid group with a side chain, essentially creating a new amino acid. So that's what you see here. We took the amine group from valine and added it to the other portions of this central carbon that would lead to a glycine. So this amine group was transferred, and that created a glycine amino acid. Right? And so again, non-essential amino acids we can synthesize, so we don't necessarily need to get them from our food, but the essential amino acids we do. And so here you can see the list of all essential amino acids. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, and then 11 non-essential. So these 9 essential amino acids we must consume from our diet. Luckily, this isn't necessarily too difficult to do and doesn't need to be something that you spend a lot of time thinking about. The exception would be if you strictly adhere to a vegetarian or vegan diet, and we'll talk about that more later. But most food sources carry at least some, if not most, of these essential amino acids, and most meats in particular will carry all 9 of our essential amino acids. So it's not too difficult to get them in our food, but not getting enough of them can lead to other defects. All right. So proteins are made in our cells with the instructions carried in our genes. And so the process of taking those instructions from our genes, from our DNA, and turning them into a protein involves 2 main steps. So we have transcription, which is creating essentially a copy of the instructions from one particular gene, right? Your DNA is continuous and long within each chromosome, but only small portions of that DNA actually code for one single gene. And one single gene usually results in a single protein. And so the small portion of DNA that contains the gene that we need to use will be unwound. We will make a copy of it called mRNA. That mRNA can then leave the nucleus. So our DNA never has to leave the nucleus. The mRNA copy will leave the nucleus. It will take those instructions to the ribosomes, and we have many, many ribosomes in our cells, either free-floating in the cytoplasm or stuck to the endoplasmic reticulum. But this mRNA will travel to a ribosome. The ribosome will then translate the instructions from that mRNA into individual amino acids. So every 3 nucleotides codes for 1 amino acid, and so the ribosome will read these 1 amino acid at a time, and tRNA will bring those amino acids, which will be bound to the next one, lengthening this polypeptide chain until we get a full sequence of amino acids that will be manipulated and structured into a functioning protein. So again, transcription is copying the instructions from DNA into mRNA, and then translation is the ribosome reading those mRNA instructions and turning that into a sequence of amino acids, which we call a polypeptide. Okay. So the genes that exist within our DNA can be activated and deactivated during different times of life, or just when they are needed and when they're not. Okay. And so when a gene is active, that DNA is available and accessible to be transcribed into mRNA. mRNA takes those instructions to the ribosome, which will then read those instructions as individual amino acids, adding them one at a time until we get a large polypeptide, which will then carry out the function of that gene. And so genes, we typically think of like the physical characteristics that come from our genes, such as like hair color, height, eye color, right? But these also are coding for our chemical characteristics. So everything that our cells are capable of doing or will do, such as controlling chemical reactions, transporting molecules from one part of the cell to the other, or in and out of cells. So proteins carry out all of the functions of the cell using the instructions from our genes. Okay. So I guess to bring up a recent example that we talked about, right, lactose intolerance. Lactose intolerance is what shows up in individuals who do not produce lactase, the enzyme needed to digest lactose. And that's because the gene responsible for producing lactase, that enzyme, has been deactivated. Right? In most humans, it is only active during the time that we are breastfeeding. And then after that, it deactivates. Right? And so since it has been deactivated, no ribosomes are creating lactase, and thus that individual can no longer digest or chemically digest the carbohydrate lactose. Okay. So during translation, the ribosome will help create bonds between individual amino acids through a process called dehydration synthesis. So essentially removing a water molecule. So it'll move a hydrogen from one and a hydroxyl group from the other. So one hydrogen and then one O and one H, one oxygen, one hydrogen. Those will be removed to create one water molecule. and that leaves one available bond for those amino acids to now bond with each other. So if we follow what's happening here, our second amino acid here is going to lose a hydrogen from its amine group. So we have a nitrogen with just one hydrogen left over. And amino acid 1 here is going to lose a full hydroxyl group, this OH, leaving an available bond from carbon to bond with the available bond from nitrogen. So you see that hydroxyl group missing here. So we remove one oxygen and one hydrogen, and then one other hydrogen, making water, H2O. Okay. And this process will continue, forming more and more peptide bonds through dehydration synthesis until we get a long chain of hundreds of amino acids that will be molded into a functional protein. And so I include this example just to show you how vast the options and differences between individual proteins can be. Right? So since we have 20 different possible amino acids that can be used in building our protein, and most proteins are typically at least 100 and up to thousands of amino acids long. If we just use some simple math to think about the different combinations that are possible, you can see how quickly we get an insane number of possible combinations from just these 20 different amino acids. So for instance, if we want to look at a 5 amino acid long polypeptide, which I don't believe there are any functional proteins that are only 5 amino acids long, but just for the sake of an example, if there are 5 amino acids in this one protein, each amino acid slot has 20 different options for which amino acid could go there. And so if we multiply 20 by 20 by 20 by 20 by 20, so 5, 20 to the 5th power, you get 3,200,000 possible combinations of amino acids and sequences from just that one small protein. So now if we look at the smallest version of your average protein that is actually functional, which would be 100 amino acids long, we do 20 to the 100th power, and we get 1.27 times. So to translate this for anyone who's not familiar with this formation, this essentially means we would have 1.27 with 130 zeros behind it. So it'd be 127 with 128 more zeros after it. And that's a number that I don't know if we even have a term for how big that is. Okay? So an insane number or an insane amount of possible combinations that can be used to create a fully functional protein. And that's why proteins do have such a vast array of functions that they perform. There's so many possible types that we could make based on the sequence and order of these amino acids. But a protein isn't actually functional until it is folded into the shape it needs to do its job. So shape and function is what ultimately ends up determining the properties of a protein and its ability to perform its job. So we have a few different levels of structure. I'll just go ahead and go to the next one to show, right? So at the top here, we have a simple sequence of amino acids. This is what would come out of the ribosome. So the ribosome has helped bond each of these amino acids together, and then it was released as a string of polypeptides. And this is considered the primary structure, the initial structure of a protein. Okay. These amino acids will each have their own chemical property to them. So any positively charged amino acids will start to form magnetic attractions with negatively charged amino acids, creating what is called a hydrogen bond. And so those hydrogen bonds will end up creating some type of shape, typically either an alpha helix like this, so like a spiral, or a pleated sheet, which is more of just like ridges. Okay. And so that is our secondary structure, which comes from those hydrogen bonds, those magnetic attractions. Then typically another protein will help bend and shape this secondary structure into its tertiary structure, so something like this. Right? So all of the various areas that have alpha helixes and pleated sheets will be bent around, and they'll form more hydrogen bonds with each other until they are held into a particular shape. And many proteins are functional in this form as their tertiary structure. However, we also have a lot of proteins that end up combining with other proteins, or technically other polypeptides in their tertiary structure, so that we get multiple chains of polypeptides working together as one functional protein. So the example here is hemoglobin. This is the protein in our red blood cells that's responsible for carrying oxygen and carbon dioxide throughout our body. And hemoglobin is composed of 4 polypeptide chains. So it's composed of 4 of these hemoglobin polypeptides. That's why we have 4 shown here, each with their own heme units, which is where the iron in our red blood cells is used to bond with oxygen and carbon dioxide and carry that molecule or that element throughout our body. So yeah. So again, ultimately, whatever shape and form this protein has been made into by its tertiary or quaternary structure is what allows it to perform its function. And so in order for proteins to maintain that structure and be able to perform their function, each protein typically has a small range, a specific range of pH and temperature that it can maintain its structure within. So when a protein is exposed to an environment that is outside of that range, that will typically cause it to denature. So it will cause all of these magnetic attractions and hydrogen bonds that are kind of holding this shape together to be disrupted, and thus it will work backwards. It will unravel from its quaternary or tertiary structure all the way back to a single line of amino acids. And so again, that process is called denaturation. So when a protein starts to unravel, it is denaturing. And so we've talked about lots of different enzymes already throughout this course, but since we're looking at protein, we'll specifically ask about a couple protein enzymes and one carbohydrate enzyme. But just to apply this idea, if we know that proteins require an optimal pH in order to function in that environment, what do you think is the optimal pH for an enzyme like amylase? Just take a second to think about it, and you can think about all of these. So I also want you to consider what the optimal pH would be for pepsin and what the optimal pH would be for protease. So take a minute to pause here and think about this. Okay. So what you want to consider when addressing a problem like this is which environments do you typically find these proteins? So for the case of salivary amylase, that is in our saliva, that's in our mouths, and our mouths are relatively neutral, right? So near that middle pH of 7. And so in order for an enzyme to function properly in that environment, it must also require that optimal pH of about neutral, right? So around 7. So amylase being in our saliva, this is a neutral environment. So amylase requires a neutral pH in order to function. So pepsin, which is an enzyme active in our stomach, activated by the hydrochloric acid in our stomach. Okay? So pepsin needs to be able to function in a very acidic environment. Right? The chyme and gastric fluid inside of our stomachs is very, very acidic because it is pumped full with hydrochloric acid. And so any enzymes that are going to function in that environment must be able to tolerate that level of pH. And so pepsin's optimal pH is very acidic. So lastly, let's think about protease. Protease is released by the pancreas into the small intestine, where it will act and chemically digest proteins within the small intestine. So we now need to think about what pH is the environment within our small intestine. So our really acidic chyme moves out of the stomach into the small intestine. Our pancreas releases bicarbonate ions to buffer that acid and make it slightly basic. So we get to a pH of around 8 or a high 7. And so protease is most likely to be, or is at its optimal functionary pH when it is in a slightly basic environment. All right, hopefully that makes sense to you all. All right, so now types of dietary proteins. So the proteins that we consume, so full protein sources, right? So meats or legumes, right? Whatever food you're consuming that contains protein in it, we would categorize as either an incomplete or a complete protein. And this is referring to whether or not they contain all 9 essential amino acids. So an incomplete protein is a protein source that does not contain all 9 essential amino acids. So on its own, it would not be sufficient as far as providing you all of the nutrients, all the protein monomers that you need to support your growth and general health. And so incomplete proteins, which do not contain all 9 essential amino acids, are considered low quality proteins. On the opposite end, a complete protein would contain all of those 9 essential amino acids. So your body would be getting all of the amino acids that it needs in order to properly function and support your overall growth and health. And so most meat sources are complete proteins. Most of the meats that we consume have all 9 essential amino acids. So if you do regularly eat meat in your diet, this is something that you probably don't need to spend any time thinking about or considering, because most of the meat you're consuming is complete, and thus you're getting the variety of amino acids that your body needs. Really, this idea comes into play for those that consume vegetarian or vegan diets that are only eating plant-based protein sources. Since there are no plant-based complete proteins or plant-derived complete proteins, you will need to combine at least 2 incomplete protein sources together to get all the essential amino acids that your body needs. And again, we'll talk more about that later. Or sorry, I don't know why I said later. We're talking about it now. Later as in the next couple slides. So in order to create a complete protein out of incomplete proteins, you would need to combine 2 or more of those incomplete proteins. And this is a process that we refer to as mutual supplementation. So again, this is the method of eating 2 or more incomplete proteins together in order to create a complete protein. So in order to get all 9 essential amino acids. And so proteins that commonly work well together to achieve this goal, proteins that can produce mutual supplementation, are known as complementary proteins. So again, these are common pairings or groups of protein sources that achieve that goal. So that will give you all 9 essential amino acids when you eat them together. And there are lots of common examples of this. So beans and rice are examples of complementary proteins. Eating these 2 sources together will give all 9 essential amino acids. Peanut butter and bread, corn tortillas and beans. And then hummus, although we consider it one food, it is a product, it is a combination of multiple sources. Hummus, being a combination of garbanzo beans, garlic, and various oils, together can be a complete protein, right? So it achieves that goal of mutual supplementation. And so again, when eating a vegan or vegetarian diet or excluding meat from your diet, you need to make sure you're getting a variety of protein sources to achieve this mutual supplementation goal. Though it shouldn't be too difficult to achieve that goal, there are lots of very common combinations of foods that are complementary. Okay. So for instance, corn and beans acting together as complementary supplements for amino acids. Corn contains the amino acids methionine, valine, threonine, phenylalanine, leucine, and isoleucine. Okay, so all of these that we see in yellow. Beans also have these top 5, valine, threonine, phenylalanine, leucine, and isoleucine, but they contain 2 more essential amino acids that corn does not, tryptophan and lysine, but they do not contain methionine, right? So we need both of these sources together in order to make sure we get the methionine from corn and the tryptophan and lysine from the beans. That makes a complementary pairing of proteins. Okay. And so just to give you some examples of common food types and the percentage of calories in that food that come from protein. So some of the highest complete proteins or the highest concentration of protein in a complete protein source would be tuna. Right? 88% of the calories in that food in tuna are coming from protein. Grass-fed beef, so 78% of that meat is coming from protein. Chicken breast has 73%. And then for our incomplete proteins, some beans and legumes actually have some of the highest concentration of protein or the highest concentration of calories coming from protein, such as soybeans, really, really high in protein, really common in our protein substitutes, right? Soy milk, soy protein powder, etc. Right? But most other plant-based sources are relatively low. Some of our leafy greens can be high, particularly spinach. Kale even has a relatively high percentage of calories from protein. But yeah. So that is all for this portion of the lecture. And I will be recording one more, going over the rest of the information about protein. So see you next one.